The effect of different wavelengths of light on the rate of photosynthesis. The purpose of this lab was to identify how the rate of photosynthesis is impacted by various wavelengths of light. The hypothesis I've come up with for this lab is that if the wavelength interval of light hitting a spinach leaf is greater than 560 nanometers or less than 520 nanometers, then the rate of photosynthesis will occur at a faster rate than a leaf within the 560 to 520 nanometer range. The variables that are involved in this lab, the independent variable, which is our light wavelength, the dependent variable, which is the time, and our control variables, which include the number of spinach discs, the temperature, as we know temperature plays a role in the rate of photosynthesis, as it can denature certain pigments that are located in the photosystems. The last control variable would be the concentration of sodium bicarbonate solution. As we know, sodium bicarbonate contains CO2, and that plays a primary role in the Calvin cycle. The materials used in this lab were a stopwatch, a 60-watt lamp, a plastic green filter, a 10 cc syringe, 2 grams of sodium bicarbonate, 100 milliliters of distilled water, a 50 milliliter beaker, 3 spinach leaves, and a hole puncher. The procedure for this lab consists of 13 steps, but to summarize, we mix 2 grams of sodium bicarbonate into 100 milliliters of distilled water to create a 2% sodium bicarbonate solution. Then we punch 10 spinach discs out of 1 spinach leaf and place those into a syringe and place the plunger on top. We place the syringe into the sodium bicarbonate, and we pulled back the plunger to fill the syringe with 10 milliliters of sodium bicarbonate. Then, holding the syringe with the tip facing the ground, we lightly pulled the plunger up and then slightly pushed the plunger down to remove the oxygen from the leaf discs in order to allow them to sink to the bottom of the syringe. Then we placed the syringe in a 50 milliliter beaker, and then in a dark room, we used a 60-watt lamp and placed that on top of both beakers. Now, one beaker we wrapped with green filter paper, and we put a green filter on top to ensure that only green light was passing through, and then one we just used normal white light and there was nothing around it. Results and lab analysis. Our results consisted of 7 of the 10 discs in the white light beaker floating to the top of the syringe within 50 minutes. The first 5 rose to the top within the first 20 minutes, and the last 2 rose up near the 50-minute mark. In the green beaker, no discs rose to the top within 50 minutes. This proved our hypothesis, which, to remind you, was that if the wavelength interval of light hitting a spinach leaf is greater than 560 nanometers or less than 520 nanometers, then the rate of photosynthesis will occur at a faster rate and the leaf discs will rise faster than a leaf within the 560 to 520 nanometer range. The beaker with no filter and just white light contains all the white wavelengths at an equal intensity, meaning the light wavelength ranges from 400 nanometers to 700 nanometers. With wavelengths of both greater than 560 nanometers to less than 520 nanometers, the leaf discs in the white light beaker were able to undergo photosynthesis at a normal rate and rise. This is because in the thylakoid membrane, where photosystem 2 will accept the light being taken in, this light will bounce around the accessory pigments to release some energy before the energy hits chlorophyll A, so it does not denature. Since chlorophyll A is a blue-green, it takes in all other wavelengths except for blue-green, as it will reflect it. Since the white light consisted of all the wavelengths, chlorophyll A was able to undergo photoexcitation and allow for 2 electrons to be passed through the membrane and reach photosystem 1. In order to give 2 electrons back to photosystem 2, the Z protein will split a water molecule in half, giving 2 electrons to photosystem 2 and resulting in 2 proteins being made, and the first product of photosynthesis, which is half of an O2 molecule. Then photosystem 1 will get photoexcited, and those electrons will allow for NADP to become reduced and turn into NADPH. This allows for the Calvin cycle to occur and create our second product of photosynthesis, which is glucose. So since the 7 spinach discs in the white light beaker rose up, this proves that photosynthesis was occurring as the oxygen was being produced. The other 3 leaves
that did not rise up could have been due to experimental errors, as I will go into later. Whereas in the green beaker, since it is only shining light within the 560 to 520 nanometer range, the chlorophyll A will reflect the light, causing it to not undergo photoexcitation. Then oxygen is not being produced from the Z protein, splitting a water molecule to replenish photosystem 2 with 2 electrons, meaning the spinach discs could not rise with no oxygen, and photosynthesis was not occurring properly. Experimental errors. During this lab, 2 experimental errors arose. The first experimental error was having to prepare each syringe separately. We had to prepare the green light beaker first due to time constraints. As we were preparing the beaker, the syringe with the discs and the sodium bicarbonate solution were sitting in the light of the classroom for a good amount of time. We feared this would kickstart photosynthesis and create the oxygen that allows for the discs to float, and our results would not be accurate. Fortunately, this error did not impact the experiment that much, as we realized this error early enough to make sure that the white light in the classroom did not affect the syringe that was supposed to go in the green beaker. We did this by storing it in a cupboard. This would allow for no light to make chlorophyll A undergo photoexcitation, meaning photosynthesis cannot occur as no electrons were passed through the thylakoid membrane. Next time, I would put it immediately into a cupboard once the syringe is prepared. The second experimental error was the concentration of sodium bicarbonate solution. We created a 2% sodium bicarbonate solution, which for this specific lab was unnecessary. We were not testing to see how the concentration of CO2 impacts the rate of photosynthesis, rather how the light wavelength does. So due to decreasing the concentration of sodium bicarbonate, we decreased the amount of CO2 available for the discs to undergo the first step in the Calvin cycle, which is carbon fixation, ultimately resulting in the production of glucose. So since the rate of photosynthesis was already slow due to the concentration of CO2, this impacted our results as it caused for the lab to take longer for no necessary reason. Next time I would use just a pure sodium bicarbonate solution, so that way the concentration of CO2 does not impact the timing of the light wavelength on the lab. Challenges of a self-made lab. One challenge we faced by creating our own lab was time management. We created the lab and we made our procedure super intricate to make sure that the results that we get are what we want. As we actually went through with the lab, we realized that we didn't really take in how much time the lab and lab prep actually needed. We found ourselves fighting against the clock and having to do aspects of the lab at lunch to ensure we get done on time. So next time, I would seriously acknowledge the time and try the different aspects of the lab well timed to make sure that the lab is actually going to fit within a 75-minute period range. Another challenge we faced while making this lab was not realizing all the different variables that come into play once a lab is actually done. These variables included different things such as temperature, lighting, the same and proper equipment, etc. So for our lab, for example, as I mentioned before, we did not account for the lighting in the classroom that may affect the rate of photosynthesis. We also did not account for using 2 different kinds of equipment, such as using 2 different kinds of syringes or 2 different kinds of lamps, which actually does play a role on the result accuracy, because one piece of equipment may work a little differently than the other, such as the syringe. One was better at sucking the air out of the spinach discs than the other, which meant the other syringe took more time, and those discs spent more time in white light. Overall, this lab has taught me 2 very important lessons. One, that photosynthesis occurs at a slower rate when the light wavelength is between 560 to 520 nanometer range. Secondly, to seriously appreciate the time and effort that my science teachers put in to ensure our labs are actually interesting and fun, but that they're also dummy-proofed for a bunch of 12th graders to actually do properly.